next speaker is Phil Jager, a colleague from Columbia, talking about the genetic architecture of microglial traits.
So I'm delighted to be here today to share some of our work that we've been working over the last few years. And as many of you know, I just moved to the neighborhood over the summer. And we actually, the main sort of theme for the talk today was going to be the genetics of microglia. Luckily for Dan Felsky, who's a talented postdoc, he's actually was picked for one of the talks. So we moved that portion of the talk into his, which is right after mine. So I'll focus on a slightly different but connected sort of set of experiments. So the challenge that we set ourselves to is the following, that we're very interested in how the aging brain deals with the various sources of variation in its function. And what I've highlighted on the right side are these different sources of variation that we want to think about and we want to really understand mechanistically. Some of these in red are the aging-related pathologies which is our primary interest. There are other very common factors that are happening in the same brain at the same time. And of course, this is another source of variation that we have to take into account. Then there is the brain reserve and resilience, meaning so the innate ability of the neurons or the neural networks to resist the pathology, but also changes in the brain that can be modified over time, such as levels of education, which influence your cognitive reserve. But mechanistically, we still don't really understand this very well, but clearly it has a big impact on brain function. And of course, all sorts of other technical confounders that some of which are known, many of which are not. We, like many other people, are generating lots of different data from these brains. And I think we're in a unique position to generate many levels of data on the same individuals, which has been really sort of the focus of our work. And the question is, how do we begin to integrate all these data together? Because ultimately, what we have here to the left are a whole host of different risk factors, some genetic, some experiential, that ultimately lead to these clinical syndromes that we face as clinicians. And of course, in between, we have a host of different intermediate phenotypes and pathways that lead us from risk factors to clinical syndromes over here. And we want to understand this process. We can do this because of two superb cohorts created by my colleague, David Bennett, in Chicago, the Religious Order Study and the Memory and Aging Project. In this case, these are Catholic priests, nuns, and brothers. These are just older men and women living in the facilities around Chicago. In both cases, the key here is that individuals are recruited into the study when they're older, but non-demented, and they sign an anatomical gift act. So they donate their brain, and in the case of MAP, also their spinal cord, selected nerves, and muscles at the time of death. There are over 3,000 participants in these studies, and over 1,300 are deceased. And so we really have an enormous sort of set of samples to explore. What's really beautiful about it is that there are annual cognitive testing in each subject, as well as neurological evaluations, with 21 different tests that are performed in each subject. Also, there is antemortem imaging, and at the time of autopsy, the brain is imaged again. So really a host of data that David has accumulated, and over the last decade, we've started to generate different molecular data from his samples, which are sort of summarized here. Again, what we're trying to do is to reconstruct the trajectory or the sequence of events from risk factors to syndromic phenotypes over here. We started off with genotypes, went on to several different layers of epigenomic data, which I won't talk much about today, RNA-seq profile, and most recently, actually, with the New York Genome Center, we generated whole genome sequence data on 1,200 of these subjects. And there are a couple of other data sets that are in the works for this year. And again, a number of different phenotypes, many of which I don't have time to get into. But that, of course, please approach me afterwards if you want to know more about them. One thing I wanted to highlight is that there are many sources of variation that we don't typically think about when we analyze these data. Last year, a colleague of mine, Andrew, and I sort of worked on really an interesting analysis where we looked at both diurnal and seasonal variation in these RNA sequence data and epigenomic data that we collected from the frontal cortex. And we can do this because, of course, people die throughout the day and throughout the year in a relatively random fashion. And so we can then use this to perform these analyses of both diurnal variation, where you can see here variation in the expression of the period gene over the course of the day, and over the course of the year, there's a similar periodicity to the expression of period 1. Now, at the genomic level, about 5 to 10% of the genome actually undergoes these diurnal or seasonal rhythms over here. And again, this is not the purpose of this talk, and we actually published this last year. But I just wanted to highlight these important sources of variation that are often not explicitly sort of measured in data. We also generated a reference atlas for the affected genetic variation on the frontal cortex in terms of the transcriptome, DNA methylation, and our ChIP-seq data in an integrated fashion. So we performed these analyses all together. And again, this was sort of published last year. So just sort of highlighting this sort of background that, again, informs our analyses going forward. But what I wanted to share with you today is our more recent work to establish a network map of the Asian cortex. And to do this, we did, as many people do, sort of collected RNA sequence data from the frontal cortices of this case. This is the initial data set of about 540 individuals. Individuals are in rows. In columns, we have the genes. And clearly, there's a pattern, there's structure to these data. And we can use co-expression patterns to define modules of genes that are co-expressed. So we did this, and actually we then explored whether these modules of coherent co-expression are present in the ChIP-seq data, and they are, as you might expect. We can also relate then these individual modules to different traits, which is done here. Each module is one row, and you have the different traits that we've looked at, such as the accumulation of amyloid and tau pathologies, two of the key pathologies for Alzheimer's disease, a pathological diagnosis of Alzheimer's disease, and the slope of global cognitive decline in each person. Again, because we have longitudinal cognitive data, we can establish person-specific trajectories of cognitive decline. How stable are these modules? So what we did is actually we repurposed a data set from our colleague Bin Zhang at Mount Sinai, where we imposed our definition of the modules that we defined and repeated the analysis in relationship to Alzheimer's disease. And these are the results for each module in our study over here on this axis. On the y-axis, the results from Bin's data that we repurposed for this purpose. And you can see that, again, the results between the two data sets are highly consistent, showing that the reproducibility of this module level analysis. So this is nice, but we want to take a step further. And so we deployed some Bayesian network structure learning approaches to try to prune the relationships between different modules and traits and modules and confounding variables to those that are most direct, and also to try to infer directionality of these associations. And of course, this is cross-sectional data by definition. These are autopsies. We can't have longitudinal data, but we can try to infer statistically the most likely direction of the association. And so this is done here, each circle here being one of the modules. Altogether, there are 47 modules that we've identified. Here are the top 11 ones. And what we prefer to do is actually to explicitly model the proportion of different cell types that are found in the cortical tissue in our network model. So these are the squares. Finally, the traits are over here. And so what we have now is basically a representation of the Asian cortex that we can then leverage for further work. And we use it really as a framework to begin to explore how different pathologies, for example, begin to alter these networks, and how, to balance this, other factors such as cognitive reserve and resilience may affect this. And of course, genetic variation has a large influence. Today, I'll focus on one thing we did with this network map, which is to begin to explore the role of the immune component of the cortical network map. So to do this, what we did is to take advantage of a different set of experiments that were happening in the lab, led by my colleague Betsy Bradshaw, who's in our center at Columbia, and an assistant professor there, and a very talented postdoc, Marta Ola, who's here today. So Marta took autopsy samples from Chicago that were flown overnight, dissected them, purified them, and eventually extracted live microglia from this autopsy tissue. And then what we did is we had a small number of individuals who had bulk level or microglial profiles, so 10 individuals from whom we had purified human microglia. And we compared the level of expression of each gene to that seen in the bulk cortex. And then what we did is to define a set of genes that we deemed to be enriched in microglia. And by definition, this was genes whose expression was fourfold increased in the microglia. Also, we had a minimal expression, sort of a threshold of expression that we imposed as well. So we actually looked at two other papers that had reported expression patterns of human microglia that were published last year. There's a lot of overlap. So there's two other papers, Galatro et Gosselin et al. These are quite different. These are actually relatively young individuals, pediatric and young adults. These are middle-aged individuals, and we have older individuals. You can see that in terms of the genes that seem to be enriched in microglia, we clearly have identified a common core of genes that are associated with microglia, but yet there's still a lot of heterogeneity amongst the different individuals, probably because of age differences for one thing, and also because each of these data sets is still relatively small. Over here, we just see some reference genes for different cell types, including ones that are expected to be in microglia and many other ones that are not, just to show that we really have relatively limited contamination from blood in our purified microglia. So one thing we did next is to see whether, how can we explore the role of microglia with our data in different diseases. And the role, particularly in the Alzheimer's disease, has been highlighted in a number of different studies in different ways. But what we did is to take a look and to see whether in the genes that we define as microglia enriched, are the Alzheimer genes that we know about more likely to be in that category? And the answer is yes. So there's enrichment for microglial genes in the Alzheimer's disease susceptibility loci. The same is true for multiple sclerosis over here. Now this is, again, we have to be a little careful because they could clearly be involved, but this could also reflect sort of myeloid pathways that are shared with peripheral monocytes, which is also the same. The same is true, of course, for Alzheimer's disease as well. We also looked at ALS genes, Parkinson's and schizophrenia genes, and while there are certain genes that are clearly highly expressed in microglia, there's no particular enrichment of in microglia for those diseases. We then also looked at our large data set of autopsy samples and tried to look at some key phenotypes. And to see how did this gene set that we defined in human microglia, age microglia, how does it relate to different key phenotypes? What's quite interesting is that there's an association with aging. So as you get older, this signature of microglia is getting greater in the level of the cortex. So whether that means that the genes are being more highly expressed or that there are more microglia in the tissue is not clear yet, but there is a relationship with aging. Very intriguingly, also, there was an association with ApoE. And when we delved a little further, we were surprised that it was not related to ApoE4, which is the haplotype that has a very strong effect on Alzheimer's risk, but rather the E2 haplotype, which is protective in the context of Alzheimer's disease. And intriguingly, this haplotype actually reduces the level of expression of these microglial genes. So in summary, what we see is that, again, there's an association with advancing age, with more expression of these human microglia genes with advancing age, but this is counterbalanced by ApoE2, and ApoE4 does not appear to have much of a role in this particular case. There's a suggestive evidence for relationship with amyloid and sex, with a relationship with amyloid, again, being suggestive, which is over here, and positive, so basically more microglia with more amyloid deposition. And this over here, this is just a number of different E2 alleles. We had 22 individuals who are homozygous for E2, which are shown over here in terms of the level of expression of the gene set as a meta feature. We've also gone on to do some single-cell sequencing of these microglia. This is data from the first dozen or so individuals that we've performed. And I just wanted to show this to highlight the heterogeneity of the cell population. So what we have here are, again, different clusters of microglia. In this particular diagram, there are approximately 16,000 cells. They're mostly microglia, which are over here. There are 16 subsets. Many of them sort of cluster on this side. These are the larger clusters. There are some smaller clusters over here, and some very small clusters of cells over here that have a different phenotype and don't appear to be microglia at this time. And we just have here different genes of interest, but which we can go into later. But again, I think the key message here is that there's a tremendous heterogeneity of this cell type, and we've only looked in this case in one region of the brain, the cortex, in terms of temporal cortices and frontal cortices. If we look across individuals, so here we have the different clusters that I just showed you in the previous slide in columns, and in rows we have each individual subject. And you can see that the cluster 1 over here really accounts for most of the microglia. And so we might think that this captures the state of the homeostatic or my microglia. And then some of these may be variations on that theme. And some of the other ones may be more specific to certain conditions. Again, because we only have about a dozen different subjects, I don't want to speculate exactly how different microglial populations might be related to different diseases or conditions. But clearly, you can see heterogeneity. We have 2 subjects, for example, that have these clusters, which are absent from the others. So again, very intriguing, and it's going to push us to do a lot more samples to really understand this heterogeneity that we were just beginning to appreciate. So getting back to our network map, how can we take advantage of the network and integrate that with these data in terms of the microglia? So what we did is actually relatively simple. We just took our profile of the aging human microglia, and we asked, well, which modules are enriched for those for the gene set? And what we found is that 5 of the 47 modules are enriched, which are shown over here. One of them is extremely enriched over here, and it's also enriched for Alzheimer genes as it happens. So this is module 116. Module 5 is another one that's quite enriched, and these, when we look at other cell types, such as astrocytes and neurons, they're really not enriched at all. So they're really quite specific for microglia. On the other hand, some of these other modules over here, while they're enriched for microglial genes, they are also enriched for astrocytic genes, suggesting that these may be shared transcriptional pathways between astrocytes and microglia, because of course astrocytes contribute to inflammatory processes. So here again, we looked at the 5 different modules that we defined and their association to different traits. Now, what was quite striking is that module 116, which is the one most enriched for microglial genes, and that's also enriched for Alzheimer genes, is not particularly strongly associated with the different phenotypes such as neuritic plaques or diffuse plaques or cognitive decline. Rather, it's associated with age, again, and module 5 and module 114 are the ones that are more strongly associated with pathology. But again, with different forms of pathology, whether it's amyloid or tau in some cases. When we repeat the network analysis in a more targeted fashion, looking at these 5 modules over here, and cognitive decline, some confounders, sex, and age over here, and 5 different measures of pathologies which are related to each other, we begin to differentiate the different transcriptional programs here and their relative roles in the aging brain, because again, these different forms of pathology, as well as vascular pathology, TP43, et cetera, are all present in these older brains. So these are all processes that are happening in parallel. They're not isolated. What we find is that module 116 is really primarily related to aging, and it's not really strongly associated with the pathologies or cognitive decline, even though it's enriched for Alzheimer genes. Again, a very intriguing and perplexing sort of question that we can go into further detail later. But again, what was interesting to us is that module 5 was associated with tau pathology, whereas these 2 modules, 113 and 114, were associated with amyloid, highlighting the point that there probably are different groups or certainly different pathways and probably different subsets of microglia that are doing different things in the brain at the same time. Another thing we can do is to take advantage of our data to try to do some modeling and to see, can we actually provide a prioritized sequence of events? So we have module 5, we have the accumulation of tau pathology, and we have cognitive decline. But what's the order? Is it tau pathology leading to more module 5 to cognitive decline, or is it module 5 tau to cognitive decline? So by doing some conditional analyses, we find that the most likely explanation, again, based on cross-sectional data, is that module 5, so the accumulation of module 5, leads to more tau pathology, which in turn, that's well known, of course, that leads to more cognitive decline. So this provides a hypothesis that we can now go into the lab and to begin to dissect in vitro some of these relationships. And we actually validated this with our ChIP-seq data as well. And again, we find the same very strong association between module 5 and tau. The last element here is, so what is module 5? It's a group of genes, about 300 different genes. But interestingly, we had another phenotype in our data set, and that was the count of morphologically defined activated microglia in the cortical tissue that was available on a subset of individuals. And Dan will go much deeper into this particular data set. But what we found is that module 5 is actually correlated with the presence of these activated microglia. So it seems to capture, this transcriptional program appears to capture the proportion of activated microglia in the cortical tissue. So what we have is, in summary, is this, where we have immune dysfunction in the older brain, but it's really in different. It's in parallel process. You have different processes, some relating to amyloid pathology, some to tau pathology, and probably other pathologies as well. So with that, I'll close, and I want to highlight in particular Betsy Bradshaw, who's been my close collaborator on all of these this work in terms of the neuroimmunology of the aging brain, and also Marta Ola, who's just promoted to being an instructor and being independent herself. Thank you.